Hello and welcome, dear students. Today we are going to discuss about the genetic polymorphism and SNPs, VNTRs and CNVs. The main objectives of today's lecture are to know about genetic polymorphisms, to study single nucleotide polymorphisms, SNPs and its types, to learn about the variable number of tandem repeats, that's VNTRs, its types, and to know about the copy number variations, that is CNVs, and to study the significance of SNPs, VNTRs and CNVs. Dear students, let's first begin with the introduction to genetic polymorphisms. Genetic polymorphism is defined as variations among chromosomes, alleles or other DNA sequences. They are the simultaneous occurrences of two or more discontinuous genotypes or alleles in a population. Polymorphisms are a type of genetic diversity within a population's gene pool. Polymorphism as related to genomics refers to the presence of two or more than two variant forms of a specific DNA sequence that can occur among different individuals or populations. The most common type of polymorphism involves variation at a single nucleotide, also called a single nucleotide polymorphism or SNP. Other polymorphisms can be much larger, involving longer stretches of DNA that can be used to map or locate genes such as those causing a disease, and they can help match two samples of DNA to determine if they come from the same source or from the different source. Genetic polymorphisms, whether studied in the form of allozymes, RFLPs, mini and microsatellite variation or DNA sequences, have become useful tools in a variety of research fields such as population genetics, evolutionary genetics, systematics and molecular phylogeny, human genetics, agricultural genetics and forensics. Genetic polymorphisms through multiple alleles at individual loci provide a mechanism to tag a gene or a piece of DNA, providing a powerful tool for a variety of investigations. These investigations cover a wide berth of objectives, including identification of genotypes in paternity and forensic studies, tracking the movement of individuals in field studies, following the progression of selection experiments in laboratory populations, mapping of quantitative loci affecting economical traits in plants and animals, mapping of disease genes in the humans, and evolutionary comparisons of DNA sequences and chromosome organization between related species. The uses of genetic polymorphisms are almost endless. Dear students, let's now discuss the types of genetic polymorphism. Just as we all look different, our genomic sequences vary significantly. These differences in our genome are known as genetic variations and are caused by various types of mechanisms such as nucleotide substitutions, insertions or deletions of nucleotides, and differences in the number of tandem repeat sequences, differences in the copy number of genomic segments, and the combination of these genes. A subset of these genetic variations is defined as genetic polymorphisms when they are observed at a frequency of 1% or higher in a certain population. A genetic polymorphism is defined as a heterozygous DNA variation present greater than 1% of the population. So DNA polymorphisms comprises three types, that is tandem repeat polymorphism or TRPs, copy number variations or CNVs, and single nucleotide polymorphism, that is SNPs. Detection of these polymorphisms can be carried out by different methods, including allele-specific PCR, restriction fragment length polymorphisms or RFLP, the microarray technique, and whole genome sequencing. Tandem repeats or repetitive genetic elements include microsatellite or STRs, that is short tandem repeats, and the mini satellites or VNTRs, that is variable number of tandem repeats, which are distinguished primarily on the basis of the size and repeat pattern. The repeated sequence in microsatellites range from 2 to 6 base, while as in VNTR it ranges from 11 to 60 base pairs. These sequences consist of several repeats of simple DNA sequence pattern, and they typically do not encode a protein or have strict requirements of size and sequence. The DNA polymorphisms are classified into 6 classes. These are first generation, that is RFLP, restriction fragment length polymorphism; second generation VNTR, that is variable number of tandem repeats or mini satellites; third generation, that is microsatellite or STRPs, that is short tandem repeat polymorphism; fourth generation, that is SNPs or single nucleotide polymorphism; fifth generation, that is CNVs, that is copy number variations; and sixth generation, that is whole genome sequencing. Dear students, let's discuss each of them one by one. First, the restriction fragment length polymorphism or RFLP. The importance of restriction fragment length polymorphism in medical research was first suggested by Botstein et al. In molecular biology, restriction fragment length polymorphism or RFLP is a technique that exploits variations in homologous DNA sequences known as polymorphisms in order to distinguish individuals, populations, or species, or to pinpoint the location of genes within a sequence. RFLP is generated by differences in the size of the DNA fragment digested by a certain restriction endonuclease due to the base substitutions at the site recognized by endonuclease. Before the discovery of polymerase chain reaction or PCR, RFLP was detected by Southern analysis and required a large amount of non-degraded genomic DNAs. RFLP patterns show codominant Mendelian inheritance, and it can help us distinguish between the parental alleles, whether the allele is of maternal or of paternal origin of the particular loci in our genome. STRs, that is short tandem repeat polymorphism, and VNTR, that is variable number of tandem repeat. Short tandem repeats or STRs are short repeated sequences of DNA that is 2 to 6 base pairs long that account for approximately 3% of the human genome. The number of repeat units is highly variable among individuals, which offers a high power of discrimination when analyzed for identification purposes. It's a widely accepted notion that STRs or short tandem repeats are non-coding in nature and are therefore not implicated in gene expression. A variable number of tandem repeats are small segments of nucleotide sequences that are repetitive. A VNTR is a location in a genome where a short nucleotide sequence is organized as a tandem repeat. These can be found on many chromosomes and often show variations in length among individuals. VNTR was first reported as one type of RFLPs by Nakamura, but they are highly polymorphic with high heterozygosity in given populations because of a wide variety of the copy number of tandem repeat DNA sequences. The main difference between VNTR and STR is the length of the repeating units of each type of tandem repeats. VNTR consists of comparatively a long repeating unit of nucleotide, usually 10 to 60 base pairs long, while as STR consists of short repeating units of nucleotides, usually 2 to 6 base pairs long. Isolation of VNTR markers was done by an extension of the report of hypervariable mini-satellite sequences by Jeffrey et al. So mini-satellite probes were identified as very variable multi-locus or multiple locus simultaneously, but each VNTR marker, also called as single locus mini-satellite marker, was identified as a single highly polymorphic locus in our genome. Because of their highly polymorphic nature, both VNTRs and mini-satellite markers were applied in forensic studies and also used in clinics to monitor recipients of bone marrow transplants. Dear students, let's now discuss what is a VNTR. VNTR, or the variable number of tandem repeats, are the repeated DNA sequences at a defined locus. The repeats are clustered together and oriented in the same direction. So individual repeats can be added or removed through replication and recombination errors. This forms alleles with different numbers of repeats. The DNA segments vary in different individuals and are hence beneficial in identifying individuals in case of a crime scene or a paternity dispute. This is known as DNA fingerprinting. The tandem repeat sequences of DNA are also termed satellite DNA. These are of three main types, that is satellite, mini-satellite and microsatellite. Dear students, we will now study the types of VNTRs. Satellite DNA, these are highly repetitive DNA sequences, and each DNA sequence consists of several thousand base pairs. A satellite can measure up to 100 million base pairs. These are found occurring in the region of heterochromatin. The Y chromosome has abundant satellites. This makes it convenient for researchers to study paternal genetic transmission in mammals. The density of the DNA is the function of its base and sequence. The satellite DNA with its highly repetitive DNA has a reduced density compared to the rest of the genome. Therefore, the name satellite DNA is coined. The satellite DNA has several biological functions. Satellite DNA is present in the centromeric and pericentromeric regions of the chromosome. It regulates the functions of the centromere. They help in the formation of heterochromatin. Satellite RNA transcripts are found in plants, vertebrates and invertebrates. Dear students, let's now discuss the mini-satellite. In a mini-satellite, each repeat ranges from 9 to 100 base pairs. It is an array of tandem repeats 500 to 300,000 base pairs long. So mini-satellites have been found in association with important features of the human genome such as gene regulation, imprinting and chromosomal fragile sites. They provide the first highly polymorphic multi-allelic markers for linkage studies. Most of the mini-satellites are GC rich that possess a strong strand symmetry. Dear students, let's now discuss another type, that is the microsatellite. The repeats are very short with 2 to 6 base pairs each. The whole array ranges from 10,000 to 100,000 base pairs long. They are therefore called short tandem repeats or STRs or simple sequence repeats. They are usually found in insect and plant chromosomes and euchromatin region of vertebrates. So microsatellites are important to population genetics because of the variable number of repeats among the individuals of a population. So microsatellite markers are inherited from both parents. Therefore, they are useful for paternity tests. Highly polymorphic loci increase our ability for parental analysis. So microsatellites are not affected by natural selection, but they are influenced by gene flow, genetic drift and mutation. Dear students, let's now discuss the importance of VNTRs. VNTRs are found on many chromosomes and vary in length among different individuals. Each variant helps in personal or parental identification. VNTRs are an important source of genetic marker RFLP, which is used in the linkage analysis of genomes. A bending pattern unique to each individual is produced by the VNTRs. VNTR has its applications in forensic science, DNA fingerprinting, and other genetics and biology research. Now let's discuss the single nucleotide polymorphisms, that is SNPs. Single nucleotide polymorphisms, frequently called SNPs or pronounced SNPs, are the most common type of genetic variation among people. Differences in single base pairs known as single nucleotide polymorphisms or SNPs are a variable class of polymorphism that can be detected by DNA sequencing, restriction fragment length polymorphisms, that is RFLP analysis, and other methods such as allele-specific PCR and allele-specific DNA hybridization. There are hundreds of thousands of SNPs loci throughout the human genome, making them especially valuable for mapping human disease genes. Each SNP represents a difference in a single DNA building block called a nucleotide. For example, a SNP may replace the nucleotide cytosine with the nucleotide thymine in a certain stretch of DNA. So SNPs occur normally throughout a person's DNA. They occur almost once in every 1,000 nucleotides on average, which means there are roughly 4 to 5 million SNPs in a person's genome. These variations occur in many individuals. To be classified as a SNP, a variant is found in at least 1% of the population. Scientists have found more than 600 million SNPs in populations around the world till date. Most commonly, SNPs are found in the DNA between genes that can act as biological markers, helping scientists locate genes that are associated with disease. When SNPs occur within a gene or in a regulatory region near a gene, they may play a more direct role in disease by affecting the gene's function. Most SNPs have no effect on health or development. Some of these genetic differences, however, have proven to be very important in the study of human health. SNPs help predict an individual's response to certain drugs, susceptibility to environmental factors such as toxins, and risk of developing diseases. SNPs can also be used to track the inheritance of disease-associated genetic variants within families. Research is ongoing to identify SNPs associated with complex diseases such as heart diseases, diabetes, and cancer. SNPs act as chromosomal tags to specific regions of DNA, and these regions can be scanned for variations that may be involved in a human disease or disorder. So SNPs found to be associated with disease may be useful for diagnostic purposes. In addition, identifying which variations are involved in altering responses to drugs could facilitate the development of personalized medicine or precision medicine. This approach to treatment is based on the concept that genetic screening for specific SNPs in a person's genome can be used to select drugs most appropriate for that individual or a group of individuals. Personalized medicine could be used to avoid potentially dangerous drug responses that are the result of altered cellular metabolism caused by a specific SNP. Dear students, let's now discuss the types of SNPs. Single nucleotide polymorphism or SNPs may fall within coding sequences of genes, non-coding regions of genes, or in the intergenic regions, that is, regions between genes. SNPs within a coding sequence do not necessarily change the amino acid sequence of the protein that is produced due to degeneracy of the genetic code. So SNPs in the coding region are of two types. One is synonymous and another is non-synonymous SNPs. Synonymous SNPs do not affect the protein sequence, while non-synonymous SNPs change the amino acid sequence of proteins. The non-synonymous SNPs are of two types again, missense and nonsense. SNPs that are not in protein coding regions may still affect gene splicing, transcription factor binding, messenger RNA degradation, or the sequence of non-coding RNA. So gene expression affected by this type of SNP is referred to as ESNP or expression SNP and may be upstream or downstream from the gene. Dear students, let's now discuss another type of genetic polymorphism, that is CNV or copy number variation. Copy number variation or CNV refers to circumstances in which the number of copies of a specific segment of DNA varies among different individual genomes. The individual variants may be short or include thousands of bases. These structural differences may have come about through duplications, deletions, or other changes and can affect long stretches of DNA. Such regions may or may not contain a gene. Change in the number of copies of small sections of our genome can have big consequences. One interesting example is a gene called amylase. This gene is important for digesting starchy foods like potatoes or grains. So scientists found copy number variations of the amylase gene in different groups of people. So people from places where starchy foods were historically very important usually have more copies of the amylase gene than people from backgrounds where starchy foods were less common. So places where meat and fish were more important parts of the diet relative to starch, this suggests that ancient people who had more copies of the amylase gene were able to get more energy from starchy foods and so were able to thrive in regions where starchy food sources were important and perhaps outcompete people in these regions that had fewer copies of the amylase gene. This is one of the many ways copy number variation can have important effects. Analysis of individual human genomes have revealed an unexpected amount of variability in human populations. The most common form of genetic variation involves small changes in the genetic code that alter a single base pair. Other types of mutations range from small insertions to large chromosomal rearrangements that can be detected cytogenetically using a microscope. So the copy number variation refers to an intermediate scale genetic change operationally defined as segments greater than 1000 base pairs in length but typically less than 5 megabases, which is the cytogenetic level of resolution. CNVs or copy number variations include both additional copies of sequence, that is duplications, and loss of genetic material, that is deletions. Because CNVs change the structure of the genome, such mutations together with inversions and translocations are collectively classified as forms of genome structural variation. Recently, scientists have come to appreciate that CNVs account for much of human variability. Sequences of the human genome, that is International Human Genome Sequencing Consortium 2001, provided the roadmap that scientists used to systematically discover CNVs. When the initial draft of the human genome sequence was completed in 2001, geneticists were surprised to find that about 5% of our genetic code consisted of redundant segments that were represented multiple times in different locations across the genome. The existence of these pieces of DNA, termed segmental duplications, suggested that large segments had changed in copy number during the last 20 million years of evolution. Today, these duplicated sequences are recognized as hotspots for CNV within the human species, and they are directly or indirectly the basis for most CNVs currently associated with disease. In 2004, researchers published 2 landmark studies of healthy individuals that showed that copy number changes involving hundreds of thousands of DNA base pairs were unexpectedly common throughout the human genome. Subsequent studies using more sensitive methods extended these results, providing evidence for the subtle variation over large portions of the human genome. So human CNVs or copy number variation is now thought to affect more base pairs than other forms of mutation. In other words, if 2 humans were compared, the number of base pairs affected by structural differences in the organization and copy number stats of DNA segments would be greater than the sum of all single base pair substitution differences. Recent evidence also suggested that changes in copy number play an important role in evolution. For example, a comparison of the human and chimpanzee genetic sequences found that copy number changes account for more differences between the 2 species than do other forms of mutations. So research also reveals that copy number changes can affect the expression of genes alter the organization of chromatin and or influence the regulation of genes in the vicinity. Dear students, let's now discuss the types of CNVs. Scientists generally assign CNVs to one of two main categories based on the length of the affected sequence. The first category includes copy number polymorphisms, that is CNPs, which are common in the general population, occurring within an overall frequency of greater than 1%. So CNPs are typically small, most are less than 10 kilobases in length, and they are often enriched for genes that encode proteins important in drug detoxification and immunity. A subset of these CNPs is highly variable with respect to copy number. As a result, different human chromosomes can have a wide range of copy numbers, example 2, 4, 5, 6, etc., for a particular set of genes. So CNPs associated with immune response genes have recently been associated with susceptibility to complex genetic diseases, including psoriasis. The second class of CNVs includes relatively rare variants that are much longer than the CNPs, ranging in size from hundreds of thousands of base pairs to over 1 million base pairs in length. Also known as microdeletions and microduplications, these variants usually have a much more recent origin with a family. These CNVs may have arisen during production of the sperm or egg that gave rise to a particular individual, or they may have been passed down for only a few generations within a family. These large and rare structural variants have been observed disproportionately in patients with mental retardation, developmental delay, schizophrenia, and autism. Their appearance in such patients has led to speculation that large and rare CNVs may be more important in neurocognitive diseases than other forms of inherited mutations, including single nucleotide substitutions. Now, dear students, we will further discuss the implications of copy number variations for heredity and diseases. Copy number variations have a number of important implications. First, CNV suggests that an individual's genetic code may not simply be the sum of the genetic contributions of the individual's two parents, because the unequal crossover events responsible for CNVs occur during the production of sperm and eggs. So children may have lost or gained additional copies of genetic information that were present in either of their parent chromosomes. Second, the extent of CNV and its association with disease has led human geneticists to consider an alternate paradigm for the genetic basis of human diseases. Instead of considering disease to be largely the result of common genetic variants that have been inherited from countless generations, geneticists now recognize that large rare structural variants of recent origin may provide the genetic basis for common diseases such as mental retardation, autism, and schizophrenia. Although these structural variants may be individually rare, collectively they may be quite common, accounting for a greater proportion of the heritable risk of disease than previously assumed. In total, these data imply that the structure of the human genome is much more dynamic and malleable than previously anticipated. Dear students, it was all about today's lecture. Hope you have understood it well. Take care. Goodbye.